Okay, here we go. Okay, we are going to ask individuals to start entering the ballroom again. We're going to get started again. Okay, we're going to get started again. If you could please take your seats. And Alan's coming. You're ready. You're good. Okay. We're just gonna go with it.
Okay, no problem.
Turn that on.
Thank you. Good morning, everyone. My name is Alan Pinto. I am a 6-foot man. I am wearing dark blue pants and a blue checkered shirt. I have glasses, short brown hair, and somewhere buried very, very, very deep, I have 6-pack abs. One disclaimer before I make my introduction of John Sholar, the CEO of jCyte, is that the last time I was asked to speak about science and research to a big crowd, I was approached after my speech, and someone addressed me as Dr. Pinto. In case Mrs. Gumport is listening, I took 11th grade AP chemistry. I very generously got a C+ and have not taken a science class since. However, 20 years ago, I met my best friend, Rebecca Alexander, who many of you know and who you'll hear from later today. She introduced me to Usher syndrome and introduced me to retinitis pigmentosa, and since then I dove in and started to learn as much as I could about the research, about the science, and what's being done to help address Usher syndrome and retinitis pigmentosa. And in that time, I found that very generously, a lot of researchers and scientists would pick up my call when I would call their labs and tell me about what they were working on. One of whom is Henry Klassen, who is the founder of jCyte, along with his partner and his wife, Jing Yang. jCyte is an extraordinarily exciting company, and even more exciting is the fact that it's being led by John Sholar, who's become a dear friend over the last couple of years. He has an extremely distinguished background. He is a decorated military veteran. He is a highly accomplished patent attorney, and over the last 2 years, he has methodically and determinedly brought forward the jCyte clinical program to the point where they are going to be recruiting for their phase 3 human trial, hopefully this fall. It's a very, very promising treatment. It is gene agnostic, so almost anyone with an RP diagnosis could potentially benefit. It's minimally invasive. It's just a simple intravitreal injection. He'll tell you more about that. And again, it is a highly, highly progressed potential treatment for this community, and it couldn't be in better hands than John's. And so I'm very pleased to introduce him. Again, John Sholar, jCyte CEO, and welcome. Thank you.
So I'm told this works better. Can everybody hear me okay? So I'm John. I'm a less than 6 foot tall, white, middle-aged male with a dad bod, and a blue suit. So I'm here today to talk to you about jCyte's program for a phase 3 pivotal trial. But first, I want to talk to you about some of the data we had before. And before I even get there, I want to thank the Usher Coalition for inviting me today. Every time I get to come to one of these events and meet the community, I learn so much, and you're all so welcoming. So thank you all very, very much for having me here today. So with that, I think there's another important point to talk about here. And it's that I think everybody knows me, knows I'm pretty blunt. I think this community's been misled a couple times about where programs were in their development. I don't want to do that today. I'm going to give you exactly where we are, exactly where the data are, and exactly what we plan to do, with the understanding that, I don't control everything. I don't control Peter Marks at FDA and, a couple other problems that I have to deal with every day, although Peter's not a problem. He's a lovely man. And, what we want to do here and why we want this trial to go forward. So we have a first-in-class platform. It's an allogeneic stem cell program. A lot of folks here have heard about stem cell, and some of that stuff about stem cells is not great, right? I think there's a big difference with where jCyte is. jCyte filed an IND in 2013, I believe, and has conducted 5 clinical trials already. Those are 5 well-controlled clinical trials with all the safety data and efficacy data and things like that. So with that background, let's go ahead and go into the slides. Guess I need to grab this slide advancer. Excuse me. So IND filed in 2013. RMAT status, for those of you who don't know, that's Regenerative Medicine Advanced Therapy status at the FDA. One of the important points about that is when I file my BLA, and we will file our BLA, the review will actually statutorily only take 6 months till we get to a decision from the agency. We've already manufactured the clinical lots for those trials. I actually personally witnessed them get picked up in a cryoport shipper and shipped out to our main site at UC Irvine last week. So we're ready to go, and we're aiming for the pivotal trial to start in Q4 of this year. So what is jCell? Again, it's an allogeneic progenitor cell, stem cell. That means it's one stem cell donation actually gets grown into, doses for 400, 500, 600 people, depending on the lot, and and it doesn't matter who it goes into. A lot of stem cell therapies, the donation comes from you and it goes right back into you. This is a different approach. We're gene agnostic as well. Obviously, that's super important. We've had many, many USH patients in the trials, and we can talk about that later. This is an intravitreal injection. So anybody who knows anything about sort of Eylea or any other of the ophthalmology drugs, this is an outpatient injection into the eye. There's no immunosuppression required. The course of treatment afterwards is 5 days of steroid eye drops. And how does it work? Well, I think, I'm actually really benefited by all the earlier speakers today, so I thank all of them. I think they kind of set me up nicely for where we are with the disease and where we are with, how we think about clinical trials. And so where we're at on RP is basically, we know if we can, provide that rod-derived cone viability factor, right? All those rods have to feed the cones. If we can do that, we can not only slow the disease down, but our data actually show that in some patients, and a significant number of patients, we can actually regain more than 3 lines of vision on the eye chart. So given that we all talked about preclinical data and animal models early on, this is Dr. Clausen's animal models from 2010, I believe. That's the RCS rat model. It's kind of the gold standard model for RP. And what you see on the left is an untreated rat eye. And what you see on the right, obviously, is that rat eye if it was given jCell. And you see, obviously, the anatomy is much more intact in that one. So this gave everyone a lot of confidence, and then we went forward. And when I say we went forward, we didn't go forward once. We've now gone forward 5 times. 160 doses of this treatment have been given to patients. 108 unique patients have been treated. Patients have been dosed in the same eye more than once and dosed in their contralateral eye, i.e. in both eyes. And again, it's been incredibly well tolerated. And basically what you see is you'll see a little red dot on the side of the sclera after the injection. That's the most common adverse event. You can also see that we started at a very low dose level and have continuously brought it up. I'll come on to talk about the trial design in a minute, but one of the most important points I found from our communications with the FDA lately is, we said, well, we've done a 3 and a 6 million cell dose before. We think we're going to take the 6 million dose forward for all the reasons you'll see in the data. And they said, well, how about you try 12? And we went, well, we'd love to try 12, but maybe 9, just to make sure that we're being careful as we go forward. So we've done all of that. You'll see there's been some, I want to be very clear, you see the serious adverse events there on the bottom right. That is viewed as a very well-tolerated profile, and even those serious adverse events were resolved very easily for those patients. So when we talk about our trials, I think really we want to talk about the phase 2b trial as the most important. As far as I know, it's the largest trial ever conducted of a new drug in RP. 83 patients were randomized across 3 sites in the US. And we took everybody over the age of 18, and we kind of took all comers on genotype. Now, when we did that, we said, okay, we're going to do this as a learning study. We're trying to understand who this drug works best in. So we didn't want to really turn anybody away. So we had some very advanced patients and some not so advanced patients. Now, this is what we saw in our data after 12 months. And this is a responder analysis, so I want to make clear what that is. So this is the percentage of patients who gained more than 15 letters, 3 lines on the eye chart. And the 6 million cell dose, you see 26% of the patients gained more than 3 lines on the eye chart in the per protocol population. Now, you also see a sham effect in there that I'm going to talk about. That's that left checkerboard. We all know people don't get better without treatment in RP. So when we saw that, we realized we had to come back and look at our data, and I'll come on to explain why. We also looked, in addition to at visual acuity, we looked at contrast sensitivity, and you see what that does between the sham and control. Kinetic visual field, which is obviously your peripheral vision, and change in mobility-related function. That's the quality of life questionnaire, which any of you who've been in a part of a trial, I'm sure, have seen before. So we saw some really significant, very encouraging data in that trial, but we had that complication of the sham effect in the phase 2b, which frankly made some problems with our statistical analysis. And some really smart people on our staff went through the data, and they actually shared the data with other companies who are working in the same area, and we all kind of came together to a really interesting conclusion. And it's that if there's a large disparity between the eyes, and the treatment's only ever given in the worst seeing eye during a trial, then those patients have a hard time actually performing the tests that get us to the clinical endpoints. So while the drug works in all of those patients, it's really just about whether the data are going to be clean coming from them in a clinical trial. So we asked ourselves, well, what happens to that sham if we do that? And the answer is it goes away. The other important point we found, and again, thanks for everybody going before because I feel like a lot of the explanation's already done for me here, is we asked ourselves, well, who else does it work best in from an anatomical standpoint? And the answer was we decided we had to make sure that patients had enough rods left to support the cones, right? So the idea here is if you have more than 8 degrees of central visual field, you have about the same number of rods and cones left. So if we can save those rods, we end up also saving the cones because they provide that rod-derived cone viability factor. So when we did that, this is what the data looked like. If we take our data and say, hey, after the fact, what if we took out the patients who had that 15-letter disparity, and what if we took out the patients who had less than 8 degrees of central visual field? This is what you see. 80% of the patients gained 2 lines on the eye chart, And you see the p-value there. That's smarter people than me will figure that out, but it means it's statistically significant. And we also saw that we had a greater, that half the patients that were still there after we took out those other ones gained 50%, gained 15 letters on the eye chart. Now again, I think from my understanding of the community, everyone's pretty happy if we can find a way to slow the disease down, and I hope every one of those therapies works. To be clear, I think we're all in this together. I want every one of these trials to be successful. But what we can show here is, hey, we're actually giving people back their vision. And we didn't look at it just as a responder analysis because I think people could say, well, that's one way to go, but what is the mean of all the patients left? This is the mean of what happens to the visual acuity in those patients. A couple points to note here, because it'll be important in a minute. You see how quickly it kind of comes back. We tested those patients at 1 month, 3 months, 6, 9, 12, right? At month 1, you already see a statistically significant increase of 10 letters for the average patient. By month 3, it's 3 lines on the eye chart. By the 12th month, it's over 3 lines on the eye chart, right? So a month after the treatment, patients were already seeing better, and that's obviously exciting to us. Now, we looked at the same thing for the secondary endpoints again, because we want to ask ourselves, well, is this just an artifact of the data, or do we have something here? And as you can see, they all go up the same way. So that leads me to the pivotal clinical trial that we're about to start. So it's going to be incredibly important for us to enroll as many patients as possible, as fast as possible, to get this to the community as fast as possible. And so largely that's why I'm here today. We're going to be agnostic to genotype, so X-linked, autosomal recessive, autosomal dominant, everyone's going to be in this trial. We want people with a BCVA between 2080 and 2800 in the study eye, and of course, to not have that interocular disparity of greater than 15 letters. Just one point on the 2080 to 2800. That's not because we don't think the drug works in people outside of that range. That's because if I'm trying to show the FDA a clinical signal, you have to have between 2080 and 2800 for me to show that 15-letter increase on the eye chart, right? Doesn't mean it doesn't work in those patients, doesn't mean we won't be able to use it in those patients. It means what we're trying to aim for is that 15-letter difference of increase in vision, and that's why. The last point on there is very, very important too, and it's another piece of information that came from our friends at the Cleveland Clinic who looked at our data. The health of the retina, i.e. the thickness of the retina, is also incredibly important to this treatment. So what we found is if your OCT measurement was more than 130 microns of thickness, you were a responder to jCyte. If it was below that, you weren't. So here's the design. 40 patients will get a 6 million cell dose, 40 patients will get an 8.8 million cell dose, and then there'll be a placebo control. I want to briefly talk about what this trial is and is not. So this trial will have a control arm. Patients, so 1/3 of the patients who enroll will not be given the active dose during the trial. With that said, much like other trials that have been discussed, the second we show that the drug works, those patients, if they choose to be, will be enrolled in the active arm immediately, right? And obviously first in line. The primary endpoint is going to be that 15-letter response rate. So while a lot of folks, and I think for good reasons, are trying to show that they're slowing the disease over multiple years, and you have to run it out multiple years if you do that just to get the data you need, we kind of swing for the fences here. So we're saying we can show a 15-letter increase in vision, 3 lines on the eye chart, in 12 months. And if we do, we'll get an approval. We basically agreed with the FDA on that point in January, went to the FDA, sat down, talked through all of this, and then we were good to go. So that's the trial design itself right there. So I see I'm out of time, and I want to make sure I hit this last point, and please do come and find me afterwards. This is the patient journey. The trial is going to happen out of Irvine, California. But that trial, understanding that's difficult for a lot of people, jCyte's going to cover the expense of travel for patients who need to come out to the site 4 times over the course of a year. So for a patient and a partner, a caregiver, we'll obviously compensate them to come out and cover all the travel costs and things like that. I like to joke with people that, yes, it's inconvenient, but I'm sorry I'm paying for 4 trips to Newport Beach in a year. I think it'll work out okay. So this is the trial design. You see you fly out 4 times, and Eric's going to pay for everybody to fly first class. And we'll go from there. But here's where we're at. So, we want everyone in here to think about being involved in the trial. There's a QR code there. There's an email there. I'm here. Come talk to me anytime you want. Please find me. I'm pretty easy to find on all of LinkedIn and all of that as well. Find the team. We're happy to answer any questions you have, and we hope to see some of you being enrolled in the trial pretty soon. I want to thank these guys, and hopefully some of them you already know. This is the Scientific Advisory Board. They largely helped us do this. You know, Dr. Panesi, I know a lot of you know too. So we're going to be looking forward to working with them on the trial as well. And Paul Sieving just joined us as well from the National Eye Institute, which we're pretty thrilled about. So with that, thank you for your time. Sorry I went a little bit over, and have a great day.